We can build people the way we want. Well, we are close to it. The all-purpose gene scissors for this and superstar of biotechnology is CRISPR-Cas9 technology, short CRISPR. The euphoria is massive. Hereditary diseases and cancer could be cured with it. And human embryos have already been altered with CRISPR, and that's what scares many people. The current state on the technology right now on Clixoom Science Fiction. Subscribe for 3 new videos a week. A warm welcome.
1, 2.
Let's start with the success stories and positive sides of CRISPR-Cas, the new all-purpose tool of genetic engineering. A quick explanation: with this method, selected genes can be inserted, switched off, or even be rewritten in a simple way. It works as simple as a pair of scissors. It was not until August 2017 that this incredible fact was revealed. We humans are able to intervene in evolution, take broken genes out of the body, implant healthy genes into the body, and thus hereditary diseases are defeated. Or much worse, we'll be able to create superhumans with a simple and effective technique. Unimaginable, but that's exactly what a US team of scientists has done on human embryos. The scientists from Portland made an attempt to fertilize healthy eggs with sperm from a donor who suffered from myocardial insufficiency. They injected not only the sperm but also corresponding CRISPR proteins into the oocytes. These were supposed to cut out the broken section, which triggers the hereditary disease, and replace it with a healthy section. And indeed, in 42 out of 58 embryos, the genetic defect was resolved. Wow, but not 100% wow, because this experiment, just like similar experiments, shows significant weaknesses, and they are not to be ignored. Specifically, something strange had happened in the experiment. Not the artificial DNA snippet of CRISPR proteins was incorporated into the embryo, but the healthy snippet from the natural egg cell, which actually serves as a template for repair. To put it in a nutshell, the scientists have no clue what they did there. Similar attempts had to be stopped early. There were too many unplanned gene mutations that could bring untold consequences. The death of the embryo, malformations, cancer, or other diseases. And that's the huge problem of CRISPR. Too much can go wrong in the truest sense of the word. Too much is changed in other parts of the genetic material, and that is quite uncontrollable. Nevertheless, the US research team's attempt shows that genetically modified children are no longer a dream for the future, but probably only a matter of time. Research teams have great hopes for the treatment of other previously incurable diseases. For example, to selectively remove excess chromosomes using gene therapy. And a Japanese experiment shows that the genetic tool can go a completely different way. Instead of the usual fine work, there was a real massacre. And yes, it's almost a miracle, because the research team has managed to pulverize a complete chromosome in the truest sense of the word. For this, they bred cells of a person with trisomy 21, also known as Down syndrome, in their laboratory. You may have heard it in biology class. In this disease, chromosome 21 is there 3 times instead of twice. So far, there is no way to treat the disease in the womb. But the experiment from Japan actually makes us hopeful that this could be possible soon, maybe even during pregnancy. Other diseases such as Klinefelter syndrome, in which an additional X chromosome is present, or the so-called XYY syndrome, could be treated as well. This result is indeed amazing, and maybe even the beginning of a new era. But realistically, this method is not really ready for use yet, because there are far too many cuts in places that are not at all wanted. It is unimaginable what would happen if the wrong chromosomes were destroyed in a living organism. In addition, efficiency has so far been too low, with a success rate of 40%. Another area of research in which CRISPR-Cas technology plays a major role is organ transplantation. Last summer, an international research team succeeded in eliminating a hurdle for transplanting porcine organs into humans. With such organs, man becomes a chimera for the hybrid being of two organisms. A great success because research teams worldwide are researching that even animal organs could help people who only have the chance to continue living through an organ donation. Even pigs could be considered as organ donors for us humans. However, in the late 1990s, viruses were identified in the genome of pigs, which could be dangerous to humans. These porcine endogenous retroviruses, PERVs for short, could cause immune deficiencies and cancer. An international scientific team has now succeeded in what many others failed before, of course, with the CRISPR method. They had created about 30 piglets that were free of these retroviruses. Now the research team plans to develop the PERV-free pig strain in order to safely and effectively transplant the pig heart into a human. The technical term for this, xenotransplantation. There are still many difficulties to overcome. So far, primates that had porcine organs put into them by genetic engineers did not survive for a long time. All of the very few attempts to transplant pig hearts to humans have invariably failed, because the human immune system fights foreign tissue, even that of other humans. The results of the study may sound exciting, but many questions remain open. For example, it remains to be seen how long the bred piglets will survive without the retroviruses in the genome. So far, you do not know for sure if you need them to survive, but what if they live on and then find a way to genetically modify the pig heart? To these whole questions comes another aspect, the moral aspect. Are pigs acceptable as spare parts for humans? And where does all this lead us? Many questions and still far too few answers, because the problem is that most people outside of the science world know too little about this technique to weigh their potential, their opportunities, and especially the risks. Although the Ethics Council is taking a position on the interference with the gene shearing in the germline, there has to be much more debate on this topic, similar to climate change or nuclear energy. We urgently need to reflect on the risks we want to take and think about future generations. And here the politics come into play, because in the medical field there are mixed feelings regarding the gene scissors that can take off. In addition, billions of research and investment funds are flowing in, which makes the euphoria even greater. So it's about time to found an official institution that can oversee and regulate the whole thing. How far will technology take us? Click here for our video on transhumanism, and do not forget to subscribe and hit the bell. Stay tuned and see you soon.